Oh no.
It's been recording.
I didn't know delete it.
Perfect.
Hi kiddos.
Hello and welcome to episode 2 of DNA and RNA. Today we're going to be talking about protein synthesis.
So basically what we're going to show you is how you go from DNA to RNA to protein.
It's kind of tough topic. Bear with us. Make sure you rewind the videos. Make sure you pause the videos to take your notes. Get your color pencils out, your pens out, your pencils out.
And let's go.
Today's topic is protein synthesis. The first thing we're going to talk about today is RNA. I know that sounds really similar to DNA, but it's not the same. Okay? RNA stands for ribonucleic acid. RNA is used by some organisms as genetic material. Additionally, RNA is single-stranded, unlike DNA, which was double-stranded. RNA, just like DNA, is made of nucleotides. However, it's made of AUGC, not ATGC. Okay, so now we ask the question, what's the job of RNA? The job of RNA is to deliver information from DNA, which is in the nucleus, to the ribosomes during protein synthesis. Protein synthesis is a process that occurs in the ribosome. It joins amino acids together to make thousands of different proteins. There are 20 different amino acids in your body that can be rearranged based on your bodily needs. Well, there are two parts to protein synthesis. The first part is called transcription. This is writing something down. The second part is translation, which sounds like exactly what you might imagine it to be, which is changing language. Let's talk about part 1 of protein synthesis, transcription. During this process, RNA makes a copy of a small section, or one gene, of DNA. The body uses the base pairing rules to make the strand of RNA. So therefore, if the DNA has an A, the RNA is U. If the DNA has a T, the RNA is A. If the DNA has a G, the RNA has a C. And if the DNA has a C, the RNA has a G. As you can tell, things look a little bit different here because in RNA, you have a U instead of a T. All right, let's do an example of transcription. So let's set the scene. First, you have your nucleus. Inside of the nucleus, you have a DNA. That DNA has a bunch of genes on it. Remember, the point of transcription is to take one gene of DNA and transcribe it into RNA. So now we're going to take one gene and we're going to do just that. To transcribe, we need to remember our base pairing rule. C goes to G, G goes to C, A goes to U, and T goes to A. All right, now that we've got our gene, let's translate it to RNA. Okay, so let's start with our first letter, which is C. That translates to G. Next, we have T. That translates to A. Next, we have G, which translates to C. After that, we have A, that translates to U. Then we have C, which translates to G. Then we have A, that translates to U. Then we have C, that translates to G. C again, that translates to G. Then we have A, that translates to U. T then translates to A. G then translates to C. And T translates to A. And notice how the RNA has a U instead of a T. That's our base pairing rule at work. All right, let's do another practice. The DNA reads TAC, TTG, CAC, TAG. So that means our RNA is going to be AUG, AAC, GUG, AUC. All right, let's do a second practice. This DNA says ACG, CAT, GCA, and TGG. Pause the video and try and do it by yourself. All right, so this is the point in the video in which I want you to pause, okay? I want you to write down all of those letters that we have up on the screen, and I want you to translate them yourself.
So remember, every time DNA has an A, you'll write a U. If the DNA has a C, you'll write a G. If it has a G, you'll write a C. And if it has a T, you'll write an A.
You are translating from DNA to RNA.
So you're doing transcription. If you were correct, you would have gotten UGC, GUA, CGU, and ACC. Now let's get into translation. During translation, RNA travels to the ribosome, and then the ribosome reads the RNA code and uses that information to join amino acids in a specific order. Then the ribosome strings all those amino acids together into a protein. The thing is, we have to figure out how exactly the ribosome knows which amino acids to put together. This is figured out by something called the codon chart. Now, we might not know what a codon is. A codon is a group of 3 RNA bases that's used to code for one single amino acid. Let's figure out how to use this codon chart. For every codon, like I said before, there are 3 letters. The first letter you find on the left side of the diagram. The second letter you find on the top of the diagram. Then you want to take the third letter and find it on the right side of the diagram. Take all 3 letters, figure out where the 3 boxes meet, and then you'll know which specific amino acid is used for that codon. For example, the codon AUU, if you find the boxes where all 3 letters match, gives you the amino acid ILE. The codon UUU, if you find the boxes where they meet, gives you the amino acid PHE. Let's take a second to see what translation actually looks like. In the ribosome, the RNA is going to fall into these 3 little pockets. From there, other RNA molecules are going to come and deliver specific amino acids based on the code that's in the RNA. Remember, for each codon or each set of 3 bases, there's going to be 1 amino acid. When these amino acids are joined together, they make a protein. Now let's see if we can put it all together. I'm going to give you the DNA strand of CCT, TAC, GCT, and TTA. I want you to pause the video, write in the RNA strand, and then use your codon chart to figure out which amino acids correspond to the RNA. Now let's check our work. If you wrote the RNA correctly, you would have gotten the strand GGA, AUG, CGA, and AAU. Bonus points if you remember to put the U at the end instead of a T. That's an example of transcription. Now let's check to see if you have your amino acids correct. If you used your chart correctly, you would have gotten GLY, followed by MET, followed by ARG, and lastly ASN. The RNA to amino acid process, remember, is translation. Now, what exactly makes protein synthesis so important? Remember that your entire body is made of protein, and without protein, your body wouldn't work at all. So let's start from the beginning. The DNA code determines your RNA code. Then your RNA code is going to determine the sequence of amino acids. That specific sequence of amino acids will give your protein a unique shape. And only if that shape is correct does the protein function correctly. So ultimately, if the DNA or any other step goes wrong, you're not going to have a functional protein, and then there's going to be something wrong in your body. Maybe you're sick, maybe something's just not working right, but ultimately, proteins that aren't shaped correctly are a really bad thing. I think we need to sum it up.
Yeah.
This is a rough thing to teach you guys online.
It's confusing.
Yeah, if you're confused, no biggie. We definitely understand. So we'll summarize.
First things first, protein synthesis, all that means is make a protein. So today we taught you how to make a protein.
And there's only 2 steps.
The first one is transcription.
The second is translation.
The second is translation. All transcription means is you are literally copying the information from the DNA into RNA, and you do this because RNA is small enough to leave the nucleus. So after that happens, it goes to the ribosome where it can be translated into amino acids.
So remember, the ribosome only knows how to read the RNA, and it reads specifically 3 bases at a time, which we call codons. Each of those codons codes for 1 single amino acid, and then when you string all the amino acids together, you make that protein.
A protein! And you synthesize protein. We hope that cleared it up. Please, please, please send us your questions because we know it's hard. We know it's confusing.
Especially that codon chart can be really confusing.
We'll attach one to your Google Classroom so you can have it on hand at all times.
Good luck, guys, and please, 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 please ask us questions. We're here for you 24/7.